morning, one and all. This is Lalita Priyanka. Today I am going to talk about genomics. The term here, genomics, generally defines genome, the complete genes which are present in the DNA. So the complete genetic studies of an organism. So genomics is an area of life science that deals with the study of genomes of a particular organism, that may be plant, animal, bacteria, anything. Right? That here the term first in the earlier 1920s by H. Winkler has identified the genome, and later on the term was actually coined by Thomas Rodker in the year of 1987. So this particularly talks about the central dogma of biology or molecular biology, where the DNA, so this is the DNA, upon transcription that gives a single stranded mRNA, and upon translating the information we'll get a protein. So this is the central dogma of biology where we are going to talk about DNA, mRNA and protein. The double-stranded DNA, upon transcription, we get single-stranded mRNA. Upon translating the information from single-stranded mRNA to, finally, we are aiming at protein. So this acts as central dogma of biology, right? So why we are stressing on these points, we have to know the complete genetic information, genome studies. So for that, next, today this genomics includes many criteria like sequencing the genomes, determining the complete set of proteins encoded by an organism. So how these particular terminologies are going to be defined, just we'll look up that. Next, they go with functioning of genes and metabolic pathways of an organism. So finally, what I'm going to say is, today we have an advanced stage where we are going to sequence the complete DNA within no time. That is called as sequencing the DNA or sequencing the complete set of genes or genome. We can call it the set of genes togetherly studying is called as genome. Where individually gene, where when we are going to talk about individual part, so that acts as a small fragment or a factor or gene. If we are going to know the complete genes, so that is called as genome, the complete whole set of genes again. So this determines the complete set of proteins. Now, ultimately, we said that here, finally, we are getting a protein, right? So now the DNA is codes for one particular protein. In between, we have a single-stranded mRNA. So that translates the information into protein, right? Here, determine the set of proteins encoded by an organism. So they codes for some protein. And we have one more criteria, that is the functioning of the genes and metabolic pathways of an organism. How these particular genes are arranged, what is its metabolic pathway, how they are coding a protein, whether they are having any kind of mutations, whether they are having any kind of resistance. So everything can be studied. So here, through the genome sequencing, we can do some human genome projects. So in generally, I'm stressing about, we are going to analyze the DNA of complete set. So that is called as human genome project. So genome is all the DNA in a cell, right? So DNA in a cell, the complete genome. Here, all the DNA and all what are the chromosomes, what it may be included. So that includes genes, intergenic genes, sequences, repeats like that, palindromic repeats, palindromic sequences. We have like that way. Specifically, it is all the DNA of an organelle. For example, you take bacteria, so it has its plasmid DNA and the bacterial DNA like that. When that is prokaryotic. Again, we come to eukaryotic. Yeast is a good example for the eukaryotes, right? Saccharomyces cerevisiae, we can quote one of the example. And all the higher organisms, they have this particular DNA in that. Eukaryotes can have two to three different types of genomes, like mitochondrial DNA, chloroplast DNA. For higher plants, I'm talking about higher plants, they have cpDNA, that is chloroplast DNA. If you talk about higher organisms, they are like animals, only, they have mitochondrial DNA, right? They have nuclear DNA, mitochondrial DNA, and plastic DNA. Clear? Which is not specific, not specified, and the genome usually refers the nuclear. Why? Because they undergoes karyokinesis. Nothing but the nucleus divides place, right? In the that is called as karyokinesis, and the cell divides is called as cytokinesis again. So So that's the traditional botany again. Next, genomic sequencing history. When you talk about the first, what they have sequenced, the first genome to be sequenced was that Haemophilus influenza, that in the year of 1995. Then later on, the Escherichia coli, E. coli stands for Escherichia coli genome, was completely sequenced in 1997. The bacterial DNA, E. coli, is a good example for bacterial DNA. Next, I have also given one of the example like yeast, Saccharomyces cerevisiae, which is eukaryotic one. So they have a base pairs of 12.8 into 10 to the power of 6, like that, base pairs, and worms like some of the other eukaryotic organisms. So they have sequenced the genomes, which is eukaryotic organisms in the year of 1919. Then this